All right. In this video, I'd like to get a discussion going about membrane potential, more specifically resting. Okay, membrane potential. And then I'd like to use this as a segue into another video to discuss the action potential. Okay, so we're going to cover a few different topics while discussing this. One, we will just start out by defining what membrane potential is. And then in order to understand membrane potential, we have to take a look at just some basics about membrane biology. And then, just basically go over diffusion real quick again, ionic concentrations, and then I want to talk a little bit how we can quantitatively figure out some forces that are involved in membrane potential called equilibrium potentials, and then we'll put it all together, summarize it all up towards the end. Okay, so on that note, let's get going. Now, the membrane potential, what it is, it's an established charge created by an unequal distribution of ions across the cell membrane. Okay, so let's say we're looking at a nerve cell. Let's say we take a section of an axon of a nerve. All right, so, and then let's say I want to take an electrode and I wanted to measure the electrical charge of this. Okay, so let's say we graph this out. Now, so then I'll take a micropipette catheter that's about 1 micrometer in diameter at the tip, and then what I'll do is I want to get ready to poke this in. All right, and then this catheter is going to be hooked up to, the voltmeter and amplifier and all that, so we can measure a charge. And if we are graphing this out, let's say this is 0. All right. While this catheter is remaining within the fluid that this nerve cell is soaked in, there will be no charge, because we're talking about humans here. Okay, essentially the electrical charge of extracellular fluid is 0. There's no charge. Okay, but now all of a sudden, let's say the tip of this electrode penetrates the cell membrane. Now what you're going to see is you're going to see a dip. Okay, you're going to see a dip. All right, a dip may go down all the way to negative 90 millivolts. Okay, and then let's say I want to continue to pass this electrode down through the cell. All right, you will see that it's going to remain negative, and then this electrode, then once I pierce through the other side of the cell, then all of a sudden you'll see it go right back up to 0 again. Okay, so what we're saying here is that there is an electrical difference between the inside of the cell versus the outside of the cell, and that electrical difference, that charge difference, is created by an unequal distribution of certain ions across the cell membrane. So what you have to start thinking, that there are different concentrations of certain ions within the intracellular fluid versus the extracellular fluid. All right, so let's talk about how we establish these concentration differences. All right, then in order to do that, we have to have a basic review of membrane biology. In order to establish a membrane potential, the cell membrane has to be permeable. Permeability is the key word here. There has to be permeability to these ions, okay? And the principal ions that we're going to focus on are sodium and potassium. Okay, sodium and potassium. So let's go back to our axon here. So we'll be taking a look at this. All right, now, so permeability. How does a cell create or alter its permeability to certain substances? Remember, it does this by using channels, because remember, anything that is non-polar, meaning non-charged essentially, cannot freely pass through the cell membrane. Okay, anything that's non-polar will pass through the cell membrane. Like a steroid hormone or a lipid-based substance. Okay, but when you take a look at sodium and potassium, you'll notice that there is a net positive charge to these. Okay, now there's other ions that are involved in this I'm going to bring up in a little while. All right, but sodium and potassium are the big ones to focus here. So being that these ions carry a charge, these cannot just freely pass in and out of the cell membrane. So membranes have gates. Membranes have gates, okay? And we'll use blue to represent a sodium gate. And we'll use red to represent a potassium gate. All right, and then there are also pumps involved with this, and we'll use the green to represent that, and that's going to be what's called the sodium-potassium pump. All right, I'll talk more about that in a little bit. Okay, so remember gates. Gates, what these gates are is essentially they're proteins. Okay, so remember they're tertiary proteins that are coiled and twisted around one another. All right, and these gates and these proteins are embedded. These are transmembrane proteins embedded within the plasma membrane. All right, and one thing you have to remember about gates is that they are highly specific. Okay, a potassium, oh dang, I got my colors mixed up. Sorry about that. A potassium gate is only going to be permeable to potassium. Okay, a sodium gate is only going to be permeable to sodium. Okay, and it has to do with the chemistry of these gates. Remember, these are proteins. These are amino acid sequences that are going to have slightly different chemistries amongst them. Okay, so essentially these gates open and close. All right, and then these pumps, the sodium-potassium pump, all right, now these operate a little differently, okay? I mean, these are essentially a gate as well, but they open and close a little differently than these. All right, but these are essentially pumping ions in one direction and ions in another direction, okay? And I'll talk about that more in depth in a little bit. All right, so these gates are permeable, okay? These gates create permeability. If we want to let's say if we want to make this cell membrane permeable to sodium, it's pretty simple then. All we have to do is open the gate, okay? If we want to make this impermeable to potassium, all we have to do is close the gate, okay? If we want the cell to become more permeable to potassium, we would just add more potassium gates in there, okay? Or if we want to make it less permeable to sodium, we may take away a sodium gate. Okay, that's essentially what cells do if they want to increase or decrease their permeability to a certain ion. Okay, to a certain charged element, is they either they use these gates and they increase or decrease their numbers. All right, so that's something to think about. And now let's work our way back to diffusion again. So remember, when we're talking about diffusion, you have to bear in mind that diffusion is essentially the movement of substances from an area of high concentration to a lower concentration. Okay, across a membrane. Across a membrane of sorts. All right. So remember let's say again we've got our axon here. We've got our cell. Let's say there were, let's say there were 140 potassium ions within the intracellular fluid. And let's say there were 140 potassium ions amongst the extracellular fluid. Okay, now just their presence alone will not facilitate diffusion. Remember, there has to be permeability. So in order for these ions to move across the cell membrane, those protein gates, those channels have to be there. All right, and let's say these gates are held wide open. Okay. Now you have to remember that these particles and molecules and ions, they're always in constant motion. Okay, they're always in constant motion. All right, and there's going to be the random movement, across cell membranes. Okay, but if there's an equal amount on both sides of the membrane, there won't be a net movement either in or out. Okay, so an equal amount of potassium will randomly work its way out and in. All right, so there will be no net diffusion taking place. Now, let's put this gate back. So now let's say there are only 5 potassium ions in the extracellular fluid. Now will there be diffusion? Will there be diffusion taking place? And if so, in what direction? Okay, so remember, from an area of high concentration to an area of low concentration, potassium will diffuse out. Okay, because remember, this is all about random motion. Okay, but if there are more potassium ions on the inside of this cell, that means there's more chances these potassium ions are going to randomly move and come in contact with this gate and then move through and out the gate, versus only the 5 potassium ions that, we're basically lowering the odds that one of these potassium ions will come in contact with this gate and travel through it. Okay, that's why, that's why molecules or particles tend to diffuse from an area of high concentration to an area of low concentration. Okay, and there are various factors that influence diffusion. Okay, there are various factors that influence diffusion. One of them is the steepness of the concentration gradient. Okay, now remember, a concentration gradient, let's erase all this here for a second. Okay, so a concentration gradient is essentially a difference in concentration across the cell membrane. Okay, so if we go back to our number, there's 140 potassium ions on the inside, 5 potassium ions on the outside. We've now established a concentration gradient, meaning there's a difference in concentration across the cell membranes. Okay, and you know that this movement is going to occur until we reach an equilibrium, and then once we reach equal concentrations across the membrane, then there will be no net movements across the membrane anymore. And the steeper the concentration gradient, the more movement will take place. So let's draw another axon here, and we'll say there's 140 potassium ions in here and 100 potassium ions here. Okay, which one has the steeper concentration gradient, meaning which one has a greater difference in the concentration of potassium across the membrane? Okay, the numbers obviously speak for themselves. We'll call this axon A, we'll call this B. Okay, obviously A is going to have a greater concentration gradient. Okay, a greater steepness in its concentration gradient. So what that means then is, let's say both of these have 2 gates. Let's say both of these have 2 potassium channels. All right, so now, let's say we open these potassium channels at the same time. That means there's going to be a faster rate of diffusion in axon A because there's a greater difference in the concentration, meaning there's going to be a lot more potassium diffusing out than there's going to be diffusing in as compared to axon B. Okay, so establishing a steep concentration gradient allows for the rapid flow of these ions across the cell membrane. Okay, and you're going to see the importance of that in a little bit. So 1, the steepness of the concentration gradient. 2, the permeability. Okay, and basically permeability, comes with the number of gates. Okay, so obviously if there are far more potassium gates in axon A versus B, and let's say we open these gates all at the same time, there's obviously more venues for potassium to diffuse out. Okay, so the number of gates and steepness of concentration gradients are very large factors that play into this. Also, you have to focus on temperature. Okay, where there's an increase in temperature, there's an increase in molecular motion, and therefore there will be an increased rate of diffusion. Vice versa, if it gets cold, okay, the cooler temperature gets, the lower the rates of diffusion. Okay. The overall size of the membrane. Okay, the larger the cell, the greater the surface area, the more rapid rate of diffusion. There's more surface area, more gates, therefore more, there's more chances for diffusion to take place. Okay, so bear in mind I know you guys know this by now, but you have to remember these basic laws of diffusion and their importance. All right. Now, the particles or the ions involved, like I said, are primarily going to be sodium and potassium. So we'll go with the axon here. Now, one thing you're going to learn when learning about human physiology is that in intracellular fluid, the primary ion within intracellular fluid is always going to be potassium. Okay, I should say the primary cation. Okay, and then the primary cation within extracellular fluid is going to be sodium. Okay, it's primarily going to be sodium. All right. And there's going to be others. There's going to be other particles involved in establishing an electrical charge as well. There's also going to be what are called fixed anions. Okay, fixed anions. Because remember when I drew that graph at the beginning of the presentation, you know, we said that as we stuck that electrode into the cell, okay, while the electrode was traveling through the extracellular fluid, there was no charge. But then once it pierced the cell membrane, it dropped down to a charge of negative 90, and then once it pierced back through the other side, then we went back to 0. Okay. All right, so now you know that potassium is a positively charged ion. So there's a lot of potassium. So the question you should have asked yourself then is, if there's a lot of potassium on the inside of the cell, why is it negatively charged? And a big part of that is because of these fixed anions. Okay, remember anions are ions with a negative charge. Cations are positively charged. Okay, and the major fixed anions that are found within cells are going to be proteins and ADP. And we'll just throw ATP on there as well. So the phosphate groups on the molecules of ATP are going to have a negative charge. There's a lot of this inside these cells. There's a lot of proteins within the intracellular fluid. Okay, so as a result, and there's going to be more of these fixed anions in comparison to the amount of potassium, hence the negative charge inside the cell. Okay, so that's something to keep in mind. All right, so sodium and potassium. Sodium and potassium. All right, now the question is then, which one of these two, I mean, no, so I forgot to mention this, why do we call these fixed? Why would we call these fixed anions? Okay, we call them fixed because they do not escape. Okay, remember, these have a negative charge. Okay, and there's no gates for protein within cell membranes, and there's no gates for ATP or ADP, so these are going to remain fixed within the intracellular fluid. All right, these are not going to escape from the inside of the cell. Whereas earlier we mentioned there's potassium and sodium gates. Okay, so now the question is, which ion, out of sodium and potassium, which one plays a larger role in establishing a membrane potential? Okay, and let's take a look at that. I'm going to kind of stick with the same slide here. Just a little bit of erasing. Gotta admit, this technology is pretty cool. Remember when I was a kid and everything was chalkboards? Granted, I kind of like chalkboards, though. Okay. So let's talk about then how these ions establish a resting charge. So let's put our axon back up here. Okay, so let's take a look at the concentrations of these ions now across the cell membrane. Okay, so when it comes to potassium, now again, before I get into this, you have to bear in mind that the numbers used are going to be very different depending on what book you're looking at. A lot of these numbers I'm giving you right now are going to be primarily coming out of Guyton's Medical Physiology. Okay, there are, if you look at Netter's Atlas of Neuroscience, Costanzo's books of physiology, Eric Kandel's Principles of Neuroscience. If you take a look at Dr. Fox's physiology book, these numbers are going to be variable, okay? Or the book that we use for my class, Mary Van Hoen's 10th edition of Anatomy and Physiology, these numbers are going to vary, okay? But the concepts will remain the same, so keep that in mind. Okay, keep that in mind. So earlier I just mentioned that when we take a look at the if we take a look at the intracellular fluid versus the extracellular fluid, there's going to be a difference in the concentrations of sodium and potassium. Okay, there's going to be a difference in the concentration of sodium and potassium. Let's put and remember, there's all these fixed anions within here as we're talking about this. Okay, so we said that within the cell, there's going to be a lot of potassium. And then outside of the cell, there's going to be a lot of sodium. Okay? Now, let's take a look at the numbers here. Let's say there are, give or take, 140 milliequivalents. Oops. Per liter of potassium in the intracellular fluid as compared to the 4 milliequivalents per liter of potassium in the extracellular fluid. Okay, so as you can see, there's a great difference between sodium and potassium here. Okay, or potassium in the intracellular and extracellular fluid. Now let's take a look at sodium. Okay, with sodium here, there will be about 142 milliequivalents per liter of potassium. Oh, goodness gracious, of sodium, I'm sorry. And then there's going to be about 14 milliequivalents of sodium in the intracellular fluid. So what are we saying here? When we're saying, so when you see this milliequivalents, just think that per liter of water, there's going to be 142 sodium ions. Okay. And then, so there's going to be 14 sodium on the inside, 142 on the inside, 140 potassium on the inside, 4 on the outside. Okay, so these numbers right here that I just put up, this is what creates this resting membrane potential. This is what creates this charge. Okay, now let's talk about how we get to these concentrations. Let's talk about how we get to these concentrations. Okay. Now, we have to go back to our basic laws of diffusion, okay? Now, remember that when it comes to the diffusion of these charged ions across the membrane, there has to be some form of permeability. And, there are going to be potassium gates. There are going to be sodium gates. So, can I have a little key going here? Okay, there are going to be sodium gates, there are going to be potassium gates. Now, let's say this is just a resting nerve. It's not sending impulses, or what we call action potentials. It's just resting. It's just kind of hanging out in its environment. When this neuron is in a resting state, it's practically impermeable to sodium, okay? And, the charge of sodium alone plays into this permeability factor because, remember, sodium just freely can't move across the lipid bilayer. Okay. Also, sodium gates remain closed. Okay. So these sodium gates are going to remain closed while the cell is just hanging out and resting. All right. Sodium gates are going to remain closed. Whereas these potassium gates are going to remain slightly open. We would say that these potassium gates are leaky. Okay. We're going to say these potassium gates are leaky. All right. Now, you understand by your basic laws of diffusion then that potassium is going, we have a permeability to potassium here. Potassium is going to leak, okay? And potassium is going to leak out. It's going to move down its concentration gradient. That's just simple diffusion, okay? Now remember, there's going to be potassium moving in as well. Okay, there's going to be a couple of reasons why potassium is moving in. One, just because of the random motion of potassium in the extracellular fluid. But two, remember, we said that this cell has a charge of about negative 90 millivolts, okay? It has a negative charge. Potassium has a positive charge, okay? So let's think about this, all right? As potassium is leaking out, the inside of the cell is going to become more negative, okay? The inside of the cell is going to become more negative, okay? So you understand your basic chemistry and how, what happens when you start taking negatively and positively charged ions or elements and getting them within proximity of each other. They're going to form an attraction between one another due to their opposing charges. So what's going to happen then, as potassium continues to diffuse out, the pushing force of diffusion will force potassium out. But we will make the cell more negative on the inside. Okay, we'll make the cell more negative on the inside. Okay, And as a result, as the inside becomes more negative, that will want us to pull potassium back in, okay, due to the differences in the charge. All right. Now, what's going to eventually happen is you're going to reach an equilibrium. All right, and you're going to reach an equilibrium when the pushing force of diffusion and the pulling forces of the opposing charges equal each other out. When that happens, okay, there will be no net movement of potassium across the cell membrane, meaning there will be an equal amount of potassium diffusing out, and there will be an equal amount of potassium being pulled back in. Okay, and then if we were to put an electrode in here and measure that, then that would be our resting membrane potential, the charge of this cell when it's at rest. Okay, and now not every cell is like this. Okay, some cells have different membrane potentials than others. Okay, and then let's talk a little bit for a second about the role of this sodium potassium pump. Okay, the sodium potassium pump is very important for a couple of reasons. Now, what the sodium potassium pump is going to be doing is it's going to be pumping potassium in and sodium out. All right, now, you know now that we're working against the concentration gradient. And since we're working against the concentration gradient, this is what we're going to call active transport. Okay, we call this active because this takes energy. Okay, this simple diffusion stuff does not require energy. All it requires, the energy created is just by the difference in the numbers. Okay, but since we're moving against the concentration gradients, that's going to take work. Okay, and work in the form of the expenditure of ATP. Now, how these sodium potassium pumps are going to work? Let me clean some of this up here a little bit. What these sodium potassium pumps are going to do is they're going to pump 3 sodium ions out for and 2 potassium ions in. Okay, so simultaneously they will be pumping sodium and potassium against the concentration gradients. That's also important for establishing a membrane potential as well. So if we accumulate any excess potassium in the extracellular fluid, we know that it can be returned back to the inside of the cell. Vice versa, we will accumulate sodium eventually on the inside of the cell. I mean, I said that potassium is the primary ion that moves in and out of the cells. Sodium will leak in once in a while. Okay, so for about, let's say, every 100 potassium ions that may leak in, 1 sodium may find its way in. Okay, or when we need to generate an impulse or an action potential, sodium gates are going to open, and the inside of the cell is going to just fill with sodium. Okay, and we have to reset ourselves back to this equilibrium. So that's the importance of these sodium potassium pumps, is these help us maintain these really help us maintain this environment. Also, they help us maintain normal osmotic pressures as well. Because one thing you're going to learn wherever when you hear me say this right now, write this down and never forget this. Wherever sodium goes, water will follow. So wherever sodium goes, water goes. Okay, so that's another very important reason we need to maintain a big difference in sodium and potassium across the cell membrane. Okay, we don't, if we have excess sodium and potassium inside the cell, that's going to draw a lot of water in, and that's going to cause the cell to swell and can damage it. But if we maintain a high amount of sodium on one side, a high amount of potassium on the other, then there won't be any net movement of water via osmosis across the membrane. Okay, so that's another important reason why we need to maintain this very unequal distribution. All right, so that is essentially how we establish a membrane potential. Now let's think about this for a second, how we can quantitatively think about this. All right, now we can essentially calculate when this net movement of potassium is going to come to a stop. Okay, when this is going to come to a stop. And we do this by using what's called the Nernst equation. We do this by using what's called the Nernst equation. Okay, now we can write out the Nernst equation like so. So what we want to do, we can calculate that voltage, we can calculate that membrane potential, okay, when there will be an equilibrium between the movement of potassium in and out. And the Nernst equation goes like so. Okay, so V is going to stand for our equilibrium potential. Okay, and this is going to be a voltage. All right. R is going to be gas constant. T is going to be temperature. I'm just going to abbreviate this. And that's going to be Kelvin. F is Faraday's constant. Okay. Z is going to be the valence of the ion that we're talking about. So essentially the charge of the ion. And then, we're going to take the inverse log of these to bring us back to base E. And then CO represents concentration outside. And then you can figure out CI would be concentration inside. Okay, so what we do, so what we're saying here with this, we can use this calculation to basically figure out when there will be, without actually having to, we can theoretically use this calculation to figure out this equilibrium potential without actually having to stick an electrode inside that neuron. Okay, now we have to do this obviously by gathering some information. Okay, so basically the gas constant, what this is saying here, what we're talking about pressures that are involved in diffusion. Temperature, obviously that's important because remember that affects rates of diffusion and will affect the amount of the ions across the cell membrane. Okay, Faraday's constant, this is just, this plays into the electrical aspects of this. I'm not going to get too in depth with that. The valence, again, the charge of the ion, and then we have to be able to measure the concentrations inside and outside of the cell. Okay, we have to measure the concentrations. Now, measuring the concentration of potassium within the cell is not that easy. Okay, there are ways we can do this. Now, let's talk about this equation now. So let's say that, let's go with the first part of this. Okay, now we're going with us and us as humans, and our normal body temperature, is 37 degrees centigrade or 98.6 degrees Fahrenheit. Okay, and then we use Faraday's constant, the pressures and all that. We take the gas constant, pressure, temperature, blah, blah, blah. Okay, now we're not going to do all that. I just don't really feel the need to go into that because that's not the part that we really need to focus on. Okay, the part we need to focus on is the second part of the equation. So when we take all these constants and we take all these constants and environmental information and put them together, okay, at a temperature of 37 degrees centigrade, all right, we will come out with 61. Okay, we'll come out with 61. Okay, so we would write this as, so to represent Z, we would write this plus or minus. Okay, because you can do this essentially for any ion, across the cell membrane. All right, and then we're going to multiply that by the inverse log of this concentration outside versus concentration inside. All right, so now simply put, all we have to do now is gather information about the concentration of these ions across the cell membrane. So do you remember how much potassium we said was there? We said there's about 140 milliequivalents per liter outside, and there's about 4 on the inside. So we would write this like so. Okay, now I'll tell you right now that being that we're basically doing an inverse log of this, it doesn't matter which one of the numbers you put in the numerator and denominator. Now, I would say you should be finicky about this because when you're in science, especially if you want to go into research, you should be kind of a hyper-anal individual. Okay, so if we calculate this out, 40. So I just gave you that, I just preached you that lecture, and then I wrote it wrong. So 4 over 140. Come out to okay. Do the log. Inverse log, we'll make it, and then we multiply that by 61, and we come out with a membrane potential of negative 94 millivolts. Okay, so according to these numbers, the membrane potential of this equilibrium potential would be negative 94 millivolts, meaning there will be no net movement of potassium across the cell membrane with these numbers. Okay, now like I said, some other books are going to be a little different. It may be 140 over 10. Okay, so if we do that, that'll give you, so if we use those concentrations, that'll give you a membrane potential of negative 90 or negative 70 millivolts. Okay, if you do 140 over 5. Okay, it gives you a membrane potential of 88. Okay, but you get the concept here. And like I said, these numbers are going to vary depending on what books you're looking at. Now, you have to bear in mind that sodium and potassium, though, are not the only ions that are going to be involved with establishing a membrane potential. Okay. So they're just going to be the primary one. So potassium is the ion that we focus on, because when the cell is at rest, that is the ion that is actually diffusing inward and in and out of the cell. Okay, and like I said, we just calculated, we just looked at how we can calculate an equilibrium potential to basically, when we say equilibrium potential, meaning the electrical charge that will be established, okay, when there's no net movement. Okay, now that's important because that's what these cells want. Okay, they don't want any, they don't need, I mean, you want to try to be at this neutrality, so when the cell's at a resting state, that's not going to mess with the osmotic pressures or any other functions of the cell. Okay. Now you have to remember that there's other ions involved as well. Whenever potassium diffuses out, some chloride will diffuse in. There will be some calcium within the cells. Okay, like I said, sodium will leak in from time to time. Okay, so because of the presence of these other cations, we would say that the average membrane potential of a resting nerve is about negative 70 millivolts. Okay. And like I said, if you alter the properties of the membrane, opening or closing gates, you're going to alter the electrical charge of the cell. Now, this is important to understand, one, because, this is just one of the more, and again, I gave you a very basic explanation of this, okay? I didn't go as in-depth in this as I could have, okay? But you have to remember, but think of the clinical importance of understanding something like this. Okay. Now, in order for the cell to be, normal and functional, establishing a normal resting membrane potential is critical. Okay. And we are able to do this, one, because we have normal electrolyte levels in the body. Okay. Whenever we have fluid problems, there's a good chance we're going to have electrolyte problems, and vice versa. Okay, and so something to think about then, whenever you see a person who is experiencing fluid and electrolyte issues, the tissues that are going to be most affected by this are the excitable tissues. Okay, the muscle tissues, the nerve tissues of the body. That's why if you see someone who's severely dehydrated, I mean, how do you see them as? They're in a dizzying daze. Okay, they almost look like they're severely drunk because of the drop in brain function. These excitable tissues that rely on not only establishing this membrane potential but changing it so they can send an impulse or so a muscle can contract. It's very important that these electrolyte levels are maintained. Okay, so now let me ask you this. There's something to think about. Okay, which of these two ions is going to present more of a hazard to a person? Will potassium imbalances or sodium imbalances be more hazardous to a person? Okay, based on everything that I just talked about, think about. I'm not going to give you the answer right now, but that's something to think about. All right. Now, the next segment I'm going to stop here, and I want to make another video then talking about the action potential, basically how a nerve takes this membrane potential, alters it, so it can communicate with other tissues. Okay. And again, if you have any questions on this, don't hesitate to ask. And good luck in your studies, folks. Keep up the hard work.